Oh, there it is. Hello, everyone. I will be presenting the strategies towards the development of reagents for quantitative antibody labeling chemistry. I am Henry Chen, and I am part of the new group at ASDRP. Oh, didn't click. Okay. So a brief outline for everyone. I will start by going over the history of bioconjugate chemistry and talk about the current applications of custom conjugated antibodies and challenges with characterization of such antibody conjugates. And I will also go over 4 novel approaches towards the quantitative labeling of antibodies. And starting off, we will see that in the current world today of therapeutics, there are various different molecules of various sizes or molecular weights. And you can see that small molecules such as aspirin have molecular weights of less than 500, and small molecules have several qualities that make them desirable to be used for therapeutics, including being soluble and stable in organic solvents, synthesizable on multi-gram scales, and being discrete in their structure. While biologics, as therapeutics, have molecular weights that are much larger than small molecules, commonly in the thousands, and are commonly only stable in aqueous buffers and are challenging to prepare on even milligram scale. They are also difficult to characterize as they are non-discrete structures. And antibody-based therapeutics have been on the rise in the past decade. As you can see on the graph, the number of antibody drug conjugates going through clinical trials, and especially late phase clinical trials, has been increasing within the past 4, 5, 6 years. And on the left, you'll see what is an antibody drug conjugate. You can see that connected to the antibody are these yellow little green bits. That represents a chemical linker that basically connects a payload to your antibody. And I'll come back to that in a second. But antibody drug conjugates in the clinic, as of now, there are several FDA-approved antibody drug conjugates for treating cancer. And yeah, and how antibody drug conjugates work clinically. The antibody is able to target antigens on the cancer cell surface, and combined with a release mechanism, they are able to deliver cytotoxic payloads to cancer cells, which kills them. Yeah. Currently FDA-approved ADCs, there are more of them. And our first strategy towards the quantitative labeling of antibodies is through repurposing biological kamikaze proteins. And this utilizes the SNAP-tag technology. And this is when a SNAP-tag enzyme, let's see if, is there laser pointer? Never mind. So on the left, you will see a SNAP-tag, which is connected to a protein of interest. In our case, happens to be an antibody. And the little bit here attacks to this spot on a, it's very, the SNAP tag reacts very selectively with this benzyl guanine substrate area here, cleaving this bond and transferring the moiety into the little pocket of the SNAP tag enzyme. And this allows us to conjugate it to antibodies in a quantitative manner. Oh, I clicked in it. Okay. So our overall SNAP tag linker synthetic route, I won't be going over it too much, as we were not the greatest of student researchers at the time of synthesis. But it starts off with a particularly dangerous step, requiring the use of lithium aluminum hydride, which is pyrophoric. It would catch fire in air and water. That's obviously not good. You have to keep your reactions air and moisture free. So following that, this synthetic route here creates or synthesizes two intermediates that combine in the final step to form our linker. And at the time of synthesis, almost every single reaction here required the use of column chromatography, which we were not particularly good at, and leading to us getting low yields and finally failing the column at the end. We're ending up with our group basically abandoning this project entirely. And as of today, it is still sitting in our cabinet. Yeah. Our next strategy towards the quantitative labeling of antibodies is through the synthesis of click reagents for the GalNAc-based protein conjugation. And through the synthetic route here, it is shown to be possible to conjugate an azide tagged sugar onto an antibody, and then the azide there will then react very selectively with the strained alkyne seen on the DBCO molecule, and the DBCO molecule can be functionalized prior to being reacted with the azide with large biomolecules such as an oligonucleotide. And the synthetic route for this linker is displayed here. It contains 7 steps, which our group members have been diligently working through the past few weeks. It starts off with the creation of an oxime on the middle ring here. The second step is a Beckmann's rearrangement, where the oxime in compound 2 is converted to an amide shown in compound 3. He didn't eat and unfortunately, due to unforeseen circumstances, step 3 was unable to be displayed. But we have step 4 here, which was completed recently. It involves the addition of compound 5 onto compound 4. And where did it? And this, due to this reaction working, we thought to ourselves, maybe we can functionalize our DBC molecule here instead of at the end of the synthesis. So we tried adding a dancyl group onto our molecule, our compound 4, which is a fluorophore, and will allow us to analyze our allows us to do fluorophore things. This is currently semi-successful, as we have 2 spots by TLC in our reaction. And TLC, thin layer chromatography, is a technique that basically separates compounds from your reaction mixture by their polarity. And that tells us that we have fluorescent molecules being created, and that's good news. And then when the reaction finishes, we should be able to determine what those molecules are. And our final strategy for the quantitative labeling of antibodies is through a monoselective lysine tagging reagent, the one shown here in the blue box, which is able to selectively react in a specific manner where in a specific region of an antibody where these 2 lysine residues shown on the left happen to be in the perfect spot where it is able to react and then become conjugated to the antibody. And this only exists in the antibody on one spot, thus making it monoselective. And then having an R group as an A side allows us to do click chemistry with it, attaching a functionalized DVCO onto the end, which is useful because we can then functionalize that DVCO with biomolecules such as DNA. And previously reported synthesis of the lysine tagging reagent has 3 steps, and the third step was particularly awful. It involves the use of thionyl chloride, which is very, very harsh on your reaction setup. It tends to melt septums, which is what you use to keep your reaction closed. If it starts opening your reaction, that's not good. We plan to improve this synthesis through our route here. It ends with a Yamaguchi reaction, which I actually ran a test reaction for and showed several side products, which is not promising, but we will remain hopeful that it will work for us. Yeah, and on our way to making this linker, we screened conditions for the first step, as the first reaction we tried did not work due to the reported reaction being unfeasible, as it requires a 66-hour reflux, and since we are school children with sleep to get, we are not able to reflux an entire 66 hours just for one reaction, so that was not attempted. We tried a 5-hour reflux in the solvent tetrahydrofuran. This gave us poor conversion. And then we tried DMSO, which gave us full conversion with side products. That was pretty good, actually. And you can see here, we analyzed our compound by NMR, which shows us that we do indeed have our product, and it's not some like random white powder. Yay! And since there was very little of the product that we got, we planned to scale up our reaction so we could have more of our compound. And this was scaled up to 5 grams. And this was done in acetone with cesium carbonate and dibromoethane. And this gave us poor conversion through several days with over 40 milliliters of dibromoethane being added to our reaction. And we knew this was not ideal, as further conversion did not appear to be happening. So we thought of adding sodium iodide and tetrabutyl ammonium iodide to fully convert into our product, as sodium bromide created in this equilibrium is not soluble in acetone, thus forcing full conversion into the iodide product. And this gave us full conversion to the iodide product. But something interesting happened after we filtered our reaction, which this was confirmed via thin layer chromatography analysis. We found that our starting material had been reforming. And here is our proposed mechanism, where an iodide attacks the iodide in our product, forming ethylene gas and iodine. And this ultimately reforms our starting material, which is not ideal. Oh, what? Yeah, this is our progress and also rationale for our 3 strategies towards the quantitative labeling of antibodies. And I would like to thank ASDRP for the amazing opportunity to conduct research at their facilities, and my group members Abhinav Chalasani, Arshia Desarkar, Emma, Njoo, Chansey, Cho, Tiffany, Jang, Harong Lee, Vrinda Iyengar, and Olivia Kwok for their contributions to this subgroup. I would like to thank our collaborators at ASDRP, Kira Yamamoto and Dr. John Wang. And last of all, I'd like to thank my advisor, Dr. Edward Njoo, for his help and insights on our research journey. Questions? I don't actually have a question slide. This is it.